Welcome to the ICU Management and Practice DigiConf Microbiome in Critical Illness. Professor Jan de Waele, Intensivist, Department of Critical Care, will moderate the session. Our panelists include Francesco Forfori, Professor of Anaesthesia and Intensive Care. Samuele Ferrari, Medical Doctor, Department of Surgical, Medical, Molecular Pathology. Nathan Klingensmith, Surgical Critical Care Fellow, Department of Surgery and Critical Care. Carmina Guitart, Intensive Care Unit Fellow, Paediatric Intensive Care Unit. Jaroslava Longitano, Consultant, Department of Anesthesiology and Critical Care Medicine. And Javier Mancilla-Galindo, Unidad de Investigación, UNAM-INC. Let us now welcome the moderator of today's DigiConf, Professor Dr. Jan de Waele.
Hello, everybody. It's good to have you all here with us today. And today's topic is the microbiome. I think the role of the microbiome in critical illness these days is now undisputed. And after decades of research, we're actually finally starting to understand this. And with the advances in microbiology we've seen in the past decades, we are now able to study the microbiome. And I think many will agree with me, many of the panelists, of course, that this holds great promise. Yet many questions remain, of course, and improving insights in disease remains critical. And at least we hope to give you a few of the answers today. We'll be covering different aspects in this field, which is, by the way, not limited to the intestinal microbiome, by the way. So I think it's time to go to the panelists and hear from them.
Thank you for the invitation. We start talking about microbiome in sepsis, and the topic is very up to date because in this screenshot you can visualize in the last 10 years how many papers have been published about this argument. And this is very important because we know that the alteration in microbiome is implicated in many diseases. And like a crosstalk between different organs. And you can have paper on cardiovascular disease, depression, in bowel, chronic inflammatory disease. So a very, very important topic. What is interesting to note is that microbiome is probably a part of a bigger puzzle in which we have the bacteria, we have the intestinal cell, and we have the immunity system below the intestinal cell. And is the interaction complex and functional between these elements that give us the gut barrier and the microbiome in its complex meaning. And what is interesting to note is that in critical patient, what we observe is the more the patient is becoming critical and the more the microbiome is changing. You can visualize in upper right side how the good bacteria, the commensal bacteria, are reducing and the bad bacteria are increasing when the patient is going worse. And this aspect is very important because, as we see before, the interaction between these elements exert an effect on the cellular, intestinal cellular, the gap junction falls, and bacteria or some products may enter in the body. And what is interesting to know is that this aspect is very, very complex. And when the bacteria enters in the body, they can move towards different organs, such as the liver. There is a very important paper now on gut-liver axis, or they can move to the lungs through the lymphatic way. And this is why often there is a very strict connection between the lung and the gut in the gut-lung axis. So it's very important to underline how the change in the microbiome will exert different and important effects on distant organs. The change in microbiome is called dysbiosis. And what is interesting to note is that many factors may provoke changing in the microbiome. The sex of the patient, the age, the nutrition, the stress, and the critical illness. But what is very interesting is that hospitalization is one of the main factors that disrupt microbiome and let the patient be exposed to a following further sepsis episodes. And probably the most important factor implied in the disruption of the gut barrier is the administration of antibiotics. Different studies have shown how the type of the antibiotic, the doses, the number of the days, and the specific classes of antibiotics are probably the most important factor able to change microbiomes. And this is an example of changing in microbiome in critical ill patient. You can see in the upper left side the different sample of a patient admitted into an ICU and a sample performed at the discharge from the ICU. And what is interesting to note is that there is a very significant change in microbiome. And many factors are related, are guilty in provoking this change. Not only antibiotics, that is probably the most important one, but also the inhibition of gastric secretion, the PPI, or the mechanical ventilation, or the administration of vasopressor that reduce the perfusion of the gut. So the interaction of this aspect probably is responsible for the changing in microbiome. And the changing let the bad bacteria grow up, and the bad bacteria and this growing up probably will damage further the barrier. And in a circle, in a vicious circle, it provoke the continuous effect of this situation. In this paper, that is one of the first many years ago, it was interesting because the changing in the microbiome, and the changing was measured as a inhibition of a particular bacteria, total obligati anaerobis, and the increase of bad bacteria like gram-positive Staphylococcus and Enterococcus. And the change in this microbiome was guilty in provoking a more incidence of sepsis or bacteremia, and in changing the outcome of the patient. But it's not so easy because the individual variability is very complex. You can visualize same disease, different patient, and what is the different microbiome of these patients. So the main difficulty we have in studying microbiome in septic patient is that we don't have a baseline, and that each patient may be different from the other one. You can visualize the change, the number are the patient, the disease is upper, and what the antibiotic administered during the recovery. And what is interesting is that in all the patients, there is a reduction in the number of bacteria. There is probably the shift toward a particular negative or bad bacteria, but it's still too difficult at the moment to have a screen of the right changing in microbiome of the single patient. In COVID patient, we observe also a change in microbiomes. You can visualize the comparison between healthy volunteer and COVID patient. And what's interesting to note is that in COVID patient appear some bacteria that were not present in the healthy population. And the alteration, the disruption in the gut barrier probably is considered one of the factors that may explain the severity of COVID disease. There is some study that found a correlation between the alteration in the disruption in the gut barrier and the severity of the disease. The more the barrier is alterated, the more the COVID disease is present. And this is probably related to different mechanisms. Some authors think that the leakage of the barrier let the virus enter into the blood, into the lymph, and so the virus may be able to start a severe immunity response, or some authors think that the virus in the blood may move to different organs and there create a replication of the virus and an organ damage. What is interesting to note is that the correlation is very important, because the immune system that is below the barrier is very, very important in controlling the body response to an infection, because often here have been released AGA immunoglobulin that move in the lung, move in other sites of the body, and so it's very important this aspect. The correlation between the severity of the COVID disease and the alteration, the disruption in gut barrier is very important, because may explain why some patients have a bad outcome, may explain, because the disruption of the gut barrier persists even after the discharge from the hospital, and may be probably responsible for the chronic heal of this patient, and may offer us a new sort of intervention to treat this disease, the manipulation of the microbiome. What is interesting to know is that at the moment we don't have any fixed screen. We should start to consider microbiome like an organ, a functional organ, and so to start to measure the dysfunction of this organ, like we do for the lung or the kidney or the liver. But it's difficult because we don't have a uniform, a typical microbiome, because each individual has its own microbiome that is related to its age, what it eats, and what if it does, if it makes sport or not, or something like that. So there is a very high variability between people, and this is probably the most difficult point we have to overcome. What we should do now is try to prevent the disruption of the microbiome, and to do that we should use very antibiotic, very careful. We should try to avoid the inhibition of gastric secretion, and we should try to administer the vasoactive drug or other drugs only if really necessary. Thank you.
Okay, thank you very much for this interesting overview. And indeed, a lot of questions, a lot of topics remain, and I'm sure many of this will come back during the discussion. I would like to remind, if you're participating in this in this conference. Please don't forget to ask your questions, use the tool, and we'll come back with questions at the end of the presentation. So it's time, I think, now to go to the next presentation. Dr. Klingensmith from Emory University, who will be speaking about the potential treatments in critical illness. Dr. Klingensmith, please, you have the floor.
Thank you very much. I really appreciate the opportunity to speak today. As you mentioned, I'm Nathan Klingensmith, one of the acute care surgery, surgical critical care fellows at Emory University and Grady Memorial Hospital in Atlanta, Georgia, in the US. Mostly what I'm going to speak about today are the potential therapeutics that we have out there, or at least we're investigating, that we can alter in the setting of critical illness among the microbiome. No disclosures. So when I think about the intestinal microbiome, and that's where most of what I'm going to focus on in this talk is the intestine. I think of it in three compartments: the epithelium, the immune system, and the microbiome. And on this Venn diagram, you can see they all touch each part and have an overlap. So one part we oftentimes think of in isolation, but they truly all impact each other. And part of some of the therapeutics as we go forward, at least some of the investigation into them, played that similar role. So as we've already heard, the intestinal microbiome for each person is individual and it functions in its own homeostasis. There's the lining, the immune cells, the bacteria that are in the lumen. But in the setting of critical illness, there is a dysbiosis or a pathobiome that's starting to be described, where we have intestinal epithelial injury, a change in the type of bacteria that are still present, a virulence change in these bacteria that are there, immune system dysfunction that allows for these bacteria to potentially invade the host. The idea of what we would like to get back to is return this patient into a setting of homeostasis. So some of the potential therapeutic avenues that are under investigation are some of the things I'll talk about: prebiotics, probiotics, fecal microbial transplant, something that's a little bit older that has some mixed data, selective digestive tract decontamination. And then I just have some even further therapeutics to talk about that are sort of pipe dreams. So starting with the prebiotics, they really function as these non-absorbable particles that can be given into the gut, into any patient truly. The most specific of which that has a lot of data coming out around it is fiber. Fiber can be split into two different categories: fermentable and non-fermentable. And these are some examples of the fermentable fiber that we could eat in a diet. Non-fermentable fiber has some data coming out, but most of what we talk about here are the fermentable type of fibers. And the importance about the administration of the fiber is what they allow the bacteria that are present to do. They're able to produce short chain fatty acids, two of which most important being acetic acid or acetate and butyrate, because a lot of the signaling pathways that the immune cells and the epithelium use come from these bacteria to get back into that homeostatic picture. So some of this, the data that's come out about this, looking at short chain fatty acids in relation to intestinal barrier function. In this study, animals were given LPS as sort of their model for sepsis. You can see that the intestinal permeability went up, but when they gave the short chain fatty acids, in this case was butyrate, the permeability went down. And they also looked at the tight junction proteins and noticed that there was recovery in the tight junction proteins, ZO1, occludin, claudin-1. Claudin-2 is a different type of protein and acts the opposite of those three, so it was down-regulated as appropriate. But you can see giving the short-chain fatty acid improved the intestinal epithelial barrier. And we know that barrier function is dysregulated in the setting of sepsis and is associated with a worsened mortality in animal models. The other aspect, as I mentioned, there's this Venn diagram that dietary fiber, when it's converted to these short-chain fatty acids, acts as a signaling molecule to immune cells, specifically regulatory T cells that express FOXP3 that are able to dampen down inflammation to reduce any further intestinal epithelial damage, but also uses a signal to stimulate IgA production in the B cells. IgA, as we know, is secreted across luminal barriers and help regulate the types of bacteria that are present in the gut and prevent pathogenic bacteria from overgrowing. So in this study, animals were given this acetate as a short-chain fatty acid, and showed that there was an improvement in their IgA response targeted to the microbiota that was in the gut lumen. These GPR43 knockout animals are animals that don't respond to acetate. So you can see that the expression of their IgA decreased compared to their wild type controls because they don't have the ability to receive that signal. And whenever for the follow-up within this, they measured the IgA in the stool and saw that when they gave to wild type animals completely, when they gave acetate, they had an appropriate response in increasing their IgA targeted at the specific microbiome bacteria that were present. If we take this a little bit more into the clinical realm, there have been some small studies that are sort of pilot trials looking at the administration of fiber. In this sort of mixed heterogeneous ICU small 20-patient pilot study, we saw that there was an increase in the abundance of bacteria that were present in the patients who received fiber compared to those who did not receive fiber. And at the same time, they measured short-chain fatty acid concentration, and they noticed that though not statistically significant, trended towards significance in this small study. More ends may have made this more significant, so this is promising and at least sort of a pilot to get things going. The other avenue being probiotics. I know that someone was going to talk about probiotics specifically in their talk, so I won't go too much into it, but I can certainly answer any questions about it as well. But probiotics are basically a group of commensal bacteria that are felt to benefit the gut and systemic health. Most of the studies have been positive. They've been small, but the problem with some of the protocols that have been used have been issues with standardization. Most of the studies don't use the same bacteria. They don't use the same route of administration, orally versus via endoscopy versus rectal. We don't have a baseline flora, just like we mentioned, everyone is an individual, so we don't know where they started from and how they moved and what that means for everybody. And then the timing, are they getting how long after sepsis, immediately when they're diagnosed, further on? It was very variable, so it's hard to interpret some of those results. Furthermore, most of these patients are getting concomitant antibiotics for the treatment of their sepsis, which can reduce any probiotic that's being induced to promote healthy changes. So it's a little bit of a mixed picture and needs to sort of be parsed out in the future. There's certainly been concern when patients are immunocompromised at baseline or from their critical illness as to giving them bacteria, if that's going to be harmful or not. And there certainly have been some reports of systemic colonization into the bloodstream of some of the probiotics that were given in some of these patients. So these aren't things that we should take seriously and be smart before we start using them as actual treatment therapies. One of the other promising avenues is fecal microbial transplant. This is taking donor stool that is felt to have a healthy microbiome, good alpha diversity, which is a lot of different bacteria within itself, and good beta diversity compared to someone else, which is the type of bacteria that are there compared to another patient's bacteria, and giving it to someone who's an unhealthy recipient. And it's already being used as part of the algorithm for recurrent C. difficile colitis infections, but hasn't really translated over into the sepsis realm very much. We have some preclinical data that looked very promising from Dr. Alberti's lab at the University of Chicago. They use a model of surgery plus sepsis where they do a partial hepatectomy and then give these animals a human pathogenic slurry into their cecum. And you can see that if they have just surgery, they all survive. But if they get this slurry, the black line goes down, their survival is significantly lower than the just surgery. With these animals, what they did was they gave them back a fecal microbial transplant, and it recovered and rescued their sepsis mortality up to the blue line here. Their control for that was an autoclaved fecal transplant to say that the bacteria and what was in the stool is what was actually causing it. And they also measured the butyrate and the short chain fatty acid in these animals and showed that the fecal transplant recovered and rescued the effect of the short chain fatty acid. So there have been some usage of fecal transplant in the ICU for critically ill patients, most of which have been limited to case reports and small studies. So this is a list of most of them that have been used outside of the setting of Clostridium difficile infection. But I want to bring your attention to this. Most of these patients were much further out from their initial septic insult, except for the last patient. It was still 7 days, but that's also still further out from the initial presentation. So some caution needs to be in line with this, especially as what I mentioned before about immunocompromised states, concomitant antibiotics, things like that, before moving towards fecal transplant. Selective gut decontamination, as opposed to adding bacteria back and promoting good health, the idea from this was attempt to remove the bad bacteria, usually and classically through non-absorbable enteral antibiotics. There have been mixed results. There have been some promising data earlier, but some of the newer meta-analyses don't really show a lot of resistance, promise, especially if there is high resistant bacteria present at the time of decontamination. And hasn't been widely used across ICUs because of the concern for inducing further resistance with leaving certain bacteria behind. So just briefly on the future therapies, things that have been discussed in the literature about bacteriophage engineering to attempt to change bacterial function, bacterial editing themselves, altering the intestinal barrier, making the tight junction stronger or weaker depending on their function, using the intestines as part of an immunization and inducing IgA to sort of prime the immune system to be ready in case there is a systemic infection. And then just the metagenome and what these bacteria do to the system and how the host responds and being able to control that influence. So I appreciate again the time to speak about this, and if there's any questions, I'll be happy to answer whenever. Thank you.
Okay, thank you very much, Dr. Klingensmith. Stick around, please, for questions, but we'll take them at the end if that's okay with you. And I'm sure many questions will come. Again, for those who are listening to this conference, please use the Q&A or chat tool if you have any questions for our panelists. Okay, but in the meantime, I think we can continue and go on to the next presentation. We have Dr. Carmina Guitart from Barcelona, Spain, who will talk on the respiratory microbiome. Please, Dr. Guitart, the floor is yours.
Good afternoon. First of all, I would like to thank the organization for letting us participate in this highly interesting webinar. My name is Carmina, and in representation of our research group, I'm going to present some ideas about what we know up to now about respiratory microbiome in pediatric patients and its relation with ventilator-associated pneumonia. Sorry. As you all know, BAP diagnosis requires 2 universal criteria: PQ admission longer than 72 hours and intubation for more than 48 hours. The CDC criteria are the most widely used in pediatrics, and these include clinical, analytical, radiological, and microbiological criteria. BAP has high morbidity and mortality, and its incidence is around 5 to 14%, despite the pneumonia zero programs. Risk factors for developing BAP depend on the patient and preventive measures conducted. Lowest age, previous comorbidities, patient severity, and endotracheal tube length are the main risk factors, but most of these are not susceptible to be changed. So the question would be, are there any preventive risk factors we could identify and modify to quit BAP and achieve pneumonia zero rates? The mechanisms for BAP development are described in 3 pathophysiological ways: direct inoculation through the endotracheal tube, oropharyngeal or stomach secretion aspiration, and less frequently through hematogenous dissemination. Strategies to prevent some risk factors follow international bundles, like general strategies, for example, airway manipulation, hand cleansing, use of cuff tubes, the head of the bed at 30-45 degrees, similar than in adults. However, other recommended measures cannot be followed in children, as the subglottic secretion aspiration, because there's no dispositif for pediatrics, or digestive tube decontamination, which is controversial in children. Those measures are targeted against the principle, but it is difficult to achieve pneumonia zero rates, especially in pediatric settings. Moreover, patients with BAP are especially sensible for infections caused by antimicrobial resistance microorganisms. Thus, proper antibiotic indication and early antibiotic stewardship are so important. Due to the high rates of BAP in pediatric patients and the difficulties in the capacity of modifying intrinsic characteristics of those patients, new approaches to this nosocomial infection are required. For that reason, and attending to the principal pathophysiological ways of BAP development, oropharyngeal microbiota may be one of the investigation attention points. It has been defined that colonization of upper airway may be the beginning of the process that can conduce to a tracheobronchitis and even to a BAP, but better understanding might explain why patients develop or not one of these infections. So, which could be the role of microbiome in BAP development? The human microbiome is the ecological community of commensal, symbiotic, and pathogenic microorganisms found in human. Focusing in respiratory microbiome, it has been described as a new factor to develop community infections and BAP in pediatrics. Not only the main pathogen microorganism, but the other commensal or pathogens. General interest about human microbiome has been increasing the last 10 years. The use of next-generation sequencing and multiomic analysis reveals new insights on the identity of microbes in the lower airways, blurring the line between commensal and pathogens. Microbes are not found in isolation. Rather, they form complex metacommunities where microbe-host and microbe-microbe interaction play important roles on the host susceptibility to pathogens. In order to analyze it, there are different types of procedures: metagenomics and analysis of specific microbiota, which is based on sequencing specific regions of the DNA genes, for example, those encoding the 16S ribosomal RNA subunit regarding bacterial microbiota. Since now on, we are going to focus on bacterial microbiota, which is the most studied and known. And here we go with the Q question. Which is the role of microbiome in BAP development? Data about mouth, nasopharyngeal, and tracheal microbiome in pediatrics is scarce but promising. Respiratory mucosa is immediately colonized after birth by mother and environmental bacteria. The colonization type might be different depending on genetic and environmental factors and changes during the first years of life. An equilibrium seems to exist between the invasive and commensal bacteria when the patient is healthy, with significantly higher bacterial microbiota diversity. If it was determined that children who develop BAP have different bacterial microbiota than children who do not, some prevention actions could be implemented as antibiotic treatment in individualized cases. If we go through some publications, Hanada et al. Did a review and they noticed that some viruses infections, as influenza, may disrupt interaction between host microbial communities and host defenses, thereby contributing to the pathogenesis of secondary bacterial pneumonia and other infections. In this longitudinal study by Miyake et al., the authors collected daily cultures at admission after 48 hours and 96 hours of endotracheal aspirates from PQ patients on ventilator support. Their results suggested that in the majority of the patients, the trachea is colonized with a causative pathogen at least 1 to 2 days before pneumonia develops. They enrolled 100 patients aged from 1 day to 14 years old, and in the 3 tracheal aspirate samples collected from each patient, they observed that in the positive tracheal culture, there was an increase for Pseudomonas aeruginosa. In 16 patients who developed BAP, they obtained 10 positive tracheal cultures, 6 for Staphylococcus, 2 for Klebsiella, 1 for Pseudomonas, and 1 for Candida. They concluded that sequential evaluation of tracheal aspirates might be useful to track changes in bacterial flora at pediatric intensive care units. 2 studies showed a decrease in bacterial diversity in the pulmonary microbiome with prolonged mechanical ventilation, with an increase in Pseudomonas and Acinetobacter species, even if another pathogen was causing the pneumonia. In this slide is shown the first one by Morani et al. This was a multi-center prospective study of children requiring mechanical ventilation support for more than 72 hours, and in which tracheal aspirates were collected daily and analyzed by sequencing of the 16S rRNA gene. They observed that in patients with prolonged mechanical ventilation, there was a decrease bacterial diversity in the pulmonary microbiome and an increase in Pseudomonas and Acinetobacter species. In this slide, the second one by Zakharkina et al., they observed that the adapted island model correctly predicts loss of diversity and increased relative abundance of well-adapted microbes during intubation and mechanical ventilation. The dysbiosis of the respiratory microbiome is more profound in patients who developed BAP than in those who do not develop pneumonia. This seems to be related positively associated with the relative abundance of Burkholderia, Staphylococcus, and Pseudomonas. Despite all this previous data, we also know that colonization does frequently not progress to pneumonia and that the traditional cultures need around 3 days to be positive. So that bacterial microbiota surveillance could also help in earlier treatment, which may allow for a shorter course of systemic antibiotics or even the use of inhaled antibiotics alone. Our group has already conducted some studies about microbiome and infectious diseases in pediatric patients. Results show 3 different nasopharyngeal microbiota patterns, which we consider really interesting. The first pattern, in which Dorsogranulum genera, Moraxella lincolni, and Corynebacterium seem to have a protective profile. This pattern was significantly associated with healthy individuals. A second pattern showed that in children with invasive pneumococcal disease, after antibiotic treatment, have a reduction of Streptococcus pneumoniae and a loss of protective profile, and a repopulation of this altered niche by oral and nosocomial bacteria. And a third pattern, in which Moraxella catharralis and Haemophilus showed a trend to be related to viral respiratory infections. Regarding microbiota and pneumonia, we are understanding that complex microbial communities exist in the upper and lower airway, and the microbe-host interactions blur the line between pathogen and commensal. However, despite multiple hypotheses, there have not been yet definitely explanations about why a patient finally develops or not BAP, even when patients have similar risk factors, pathologies, or colonization. So that, if new sequencing methods were useful to diagnose nasopharyngeal bacterial microbiome pattern for BAP developing, a specific panel could be defined and developed. And moreover, this research may also allow translating the methodology to other causes of nosocomial infections, as central line catheter-associated infections or urinary tract infections associated with urinary catheter. To sum up, we would like to say that microbial community of the lung may play an important role in pneumonia, impacting susceptibility and the natural history of disease, that determining the influence of nasopharyngeal bacterial microbiota in the development of BAP in pediatric critically ill patients may improve BAP prevention, and with all this, we may allow us to reach zero pneumonia rates. Thank you very much for your attention.
Thank you very much, Dr. Guitart. That was very interesting, and clearly microbiome is not limited to the intestinal microbiome, but also respiratory and probably other parts of our body have a microbiome that we should be taking into account. Again, any questions from the audience? We'll take them at the end of the next and last presentation. Today we go to Mexico now, and I would like to invite Dr. Javier Mancilla-Galindo to talk to us on the dysbiosis COVID-19 dilemma. Javier, the floor is yours, please.
Thank you. Also, thank you very much for your kind introduction, Dr. Janteval. And I would like to congratulate all panelists for their great presentations. I believe that they have helped us to understand why the microbiome is so important for critically ill patients. And so I will be spending the following few minutes presenting to you a seemingly simple yet troubling concept, which I have named the dysbiosis COVID-19 dilemma. And this idea arose when we are trying to unravel how a few of the many pieces of SARS-CoV-2 infection could be fitted together in order to have a better understanding of pathogenesis, but especially why some patients experience disease progression while others do not. And after this attempt, we were left with more questions than answers. So I am expecting to share with you some of these questions which need to be solved to fully understand how the microbiome interacts with the host. But most importantly, if we could use some of these concepts to our advantage to provide better care for patients with COVID-19 and for critically ill patients in the ICU. And just a few declarations and disclaimers. I have no potential conflicts of interest for this presentation, and this content is for educational purposes only. It is no medical advice. Rather than leaving my acknowledgements for the end of my presentation, I would like to start by acknowledging everyone who made this possible. And first of all, I would like to thank my team for the huge efforts in bringing together this work. Victor Bolaños Toscano is a dietitian and nutritional scientist, and he is currently doing his PhD in public health and nutrition. Doctors Sofia Martinez Vasquez and Ashwin Kamar Garcia are also dietitians and nutritional scientists, and they have a large experience in providing care for critically ill patients, and most importantly, providing nutritional advice for these patients. And Liliana Fernandez Urrutia, she's a medical student who is very enthusiastic about evidence-based medicine and humanization of patient care. So thank you for bringing together this work. Also, I would like to thank Dr. Orlando Perez Nieto, since he made possible that we could have this contact with ICU management and practice to contribute to this issue. And his group wrote a very nice paper about Clostriditis difficile infection, which I invite you to read. And lastly, I would like to thank ICU management and practice since they have allowed young physicians and early career scientists like myself and my collaborators to contribute to this issue, but most importantly, to have a voice in the critical care community. So I will be spending these few minutes trying to explain this figure. We wrote about both the role of the microbiome and nutritional therapy in critical COVID-19. However, I think this is one of the most troublesome problems that we need to be thinking about, when we are studying the role of the microbiome in disease, not only COVID-19, but also for other acute and chronic diseases. And this is particularly relevant since patients with critical COVID-19 and other acute and chronic conditions are known to have alterations in their microbiomes, or the more correct term, which is dysbiosis, which refers to these alterations in patients' microbiomes, which have the potential to be harmful. And so most of this started when we were writing a paper to understand the potential mechanisms by which SARS-CoV-2 could be having an impact on inflammation and the human microbiome, but also what could be the interplay between inflammation, the microbiome, and SARS-CoV-2 infection. And so we knew from earlier research that acute viral and bacterial lung infections are associated with important disruptions of the microbiome, but also with the risk of experiencing unregulated inflammation. Preliminary studies at the beginning of the pandemic were already trying to characterize inflammation and microbiome alterations in patients with COVID-19. Dr. Francesco shared some of these studies with us. And however, there are still some things which have not been addressed by these studies. And so in this paper, we learned a few things about these interactions between the immune system, the microbiome, and the host. But most importantly, we learned that SARS-CoV-2 is highly efficient at establishing infection and evading the immune innate response. Also, immune-mediated injury is the predominant pathophysiological driver in severe and critical COVID-19. Particularly, SARS-CoV-2 is really good at avoiding the initiation of efficient interferon responses, particularly type 1 and type 3 interferon responses. But also unregulated angiotensin II may promote inflammation pathways. And this is very important because many of the comorbidities that are associated with COVID-19 progression are known to have angiotensin II and renin-angiotensin-aldosterone system alterations. And another interesting thing is that SARS-CoV-2 is able to productively infect human enterocytes. This is, well, we have seen that patients who have diarrhea, for instance, in the context of COVID-19 are at a higher risk of having disease progression. But also this active or productive infection of human enterocytes is able to dysregulate ACE2 expression, cytokine production, but also interferon pathways involved in resolution of viral infections. And so the gut microbiome is particularly important, as some of the panelists have already said. It's not only the gut microbiome, it's the entire human microbiome. But the microbiome is able to communicate and to have interrelationships with the host through many different processes. And so the microbial hosts are able to produce microbial fragments. Microorganisms are even able to translocate and travel to distant organs. They are able to produce metabolites. And since microorganisms in the gut microbiota could potentially cross the intestinal mucosal barrier, there is a potential pathway to reach the lungs, which is their access to the lymphatic mesentery system, but also the bloodstream. And there have been studies in critically ill patients and patients with pneumonia, which have found that in conditions of disease, the microbiome of the lungs tends to be similar to the microbiome of the gut. But also there are microRNAs which can travel long distances, and this could be another way by which the microbiome is communicating with distant organs. And so we also learned that few studies have aimed to characterize the microbiota, but through taxonomic characterization of their microbial components. Few studies have attempted to elucidate metagenomics or transcriptomics approaches to understand these interactions in the context of COVID-19. And we were left with some of these ideas. Establish dysbiosis in patients with chronic diseases is known to be a relevant aspect of chronic diseases like hypertension. So we see that patients with these comorbidities are at higher risk of disease progression. And we thought that studying dietary and lifestyle habits in persons with these comorbidities before COVID-19 infection could be an interesting way to establish this connection. And so we propose that cohorts of patients with chronic diseases in whom microbiota composition has been prospectively studied could be the perfect group to study how the microbiome is affecting COVID-19 progression. And so these were the key messages. Many studies have found correlations and associations between COVID-19 and dysbiosis. However, few studies have tried to investigate their directionality and potential causality. And so this reminded us of the famous chicken and egg dilemma. And despite being a very simple question, this dilemma has even required or brought quantum physics approaches to try to solve it. And we thought that some of these problems could be thought of in a similar way than the chicken and egg dilemma. So what was first, dysbiosis or inflammation? What was first, dysbiosis or critical COVID-19? So we know very little to date about the directionality of these associations, which could also be bidirectional. They don't really need to be unidirectional. And so we are left with all these individual dilemmas. And so what we tried to do was to bring this together and to represent their potential interactions and directionality, which we still need to elucidate. But why is it so important to solve these questions? So if we don't try to solve these questions first, we could find ourselves trying to solve problems which we do not fully understand yet. So this trial was recently published in September 21 of this year. And more than 2,000 patients in ICUs from more than 44 centers were randomized to receive placebo or probiotic. And they took a look to see if probiotics were able to reduce the development of ventilator-associated pneumonia. And no differences were found between patients who received probiotics and those who received placebo, which does not necessarily mean that probiotics are not useful for patients with critical disease. Maybe their benefits will be seen after a longer follow-up, or maybe we should be taking a look at other long-term outcomes. And I think that Dr. Klingensmith gave a nice background and a nice explanation of why probiotics are so troublesome to be investigated in the context of critically ill patients. But also the rehabilitation of the microbiome through a single intervention may not be the only answer. Maybe we should be starting to think about multiple interventions, which is why we try to explain in our paper. Maybe it's more about the holistic approach to the ICU patient, nutritional interventions, rational antimicrobial use, and early rehabilitation could be more important than giving patients just a single probiotic pill. And well, thank you very much for inviting me and for your attention.
Thank you very much, Javier. As often happens, when people are excited about a topic, we lose track of time. Still, I hope we have some time to maybe discuss a few things, and I hope we can get inputs from the whole panel today. Maybe I can start off with a question that also actually came up on the chat, and it's about probiotics. And based on what we hear and what we intuitively feel, maybe people would be rushing towards probiotics. What is the opinion of the panel? Should we be doing this, or do we need to take it easy and first wait for the larger studies on this? Maybe Dr. Klingensmith, maybe you mentioned probiotics, maybe you can start.
Yeah, certainly. I think that to some of the point that we've all talked about, that rushing in to use a single bacteria as our probiotic may not have the effects that we're looking for. And there needs to be some better understanding of how we're going to use not even just one, but maybe a combination, a set of specific bacteria to achieve certain goals in the ICU. And the routine use of probiotics for all patients, I would caution against at this point until we have some better studies.
Similar opinions among the other panelists?
I agree with Nathan. I think that the main difficulty is that at the moment we don't know the baseline of our patient. So adding something may work or may not work, but we don't know. If we could measure microbiome at the entering in the ICU and having a panel, and so deciding what to do during the course of disease should be better. And so we should try to focus on this aspect, the studying of something like a microbiome index or microbiome screening tool, something like that.
Let's not forget also that in patients with acute pancreatitis, there's been a study that did not specifically look into the microbiome. It didn't investigate it, but they did use probiotics, which actually associated with increased mortality for reasons unknown. But this really was a signal that we should be careful and maybe try to better understand first what actually is happening in our patients. Carmina, what is your opinion on this?
Yeah, I completely agree with all of you, because I think that firstly we need to know exactly what are we made of, what are our microbiome made of, and afterwards see how it changes when the patient needs admitted to the PICU or ECU. Mm-hmm. And afterwards see what can we do to improve their microbiome changes, maybe due to the ventilator or due to the antibiotics or other factors that can influence.
Yeah, there's also a question regarding supplements on the chat. Maybe Javier, can you comment on that? Can you elaborate whether we should be using any supplements, trying to somehow modulate the microbiome? Are we ready for that?
Yeah, so similar to the previous question, I think that we still need better evidence. I don't think we are at a point to start recommending widespread use of food supplements. And so I would say that, well, I would like to reinforce this idea that there are other interventions and more than one intervention which may be useful for patients with critical disease. Instead of focusing on supplementation or the use of probiotics, I think we are at a point right now where we could be focusing on nutritional intervention with an optimal initiation of enteral nutrition and also limitation of the use of antibiotics.
Okay, thank you very much for that, Javier. Now, I'm afraid for the sake of time, we will have to close the conference here. I think we learned a lot today from different aspects. We learned about the impact of the microbiome and it may have on several diseases. Sepsis, of course, is one of the first that we think of, and also in COVID-19, I think several of you emphasized that this is an important thing to consider. Also worth remembering, I think, is that a lot of the things we do now routinely in the ICU may affect the microbiome, and it's not limited to antibiotics, it's not limited to nutrition, but also mechanical ventilation and other interventions do affect the microbiome. So that's already an important thing, I think, to keep in mind. There are some treatments coming to us, as Dr. Klingensmith explained, many of them intuitively attractive, but probably not yet ready for large-scale use, for study, yes, but not for large-scale use. I think was a great talk by Dr. Guitart, highlighting that not only in the intestines we have a microbiome, but also in the respiratory tract, and this link to infections there definitely needs more research, and it's certainly an exciting topic. And lastly, of course, I have to thank Dr. Mancilla-Galindo, highlighting the importance of the microbiome in COVID-19. So again, thank you, everybody. Thank you for your contributions. I think it was very interesting. To thank the audience, of course, also for being here. Thank you and bye-bye.
Thank you to you. Bye-bye.
Thank you very much.
Thank you.